Cool. Thank you so much, Gary, for introduction. So hi, as Jerry introduced, my name is Suhn Rhie. I'm an assistant professor here in the Department of Biochemistry and Molecular Medicine at University of Southern California. Thank you, Active Motif, for organizing this very nice conference. And it's my honor to present our research work here. And I'd like to hear some feedback and also be more interactive through today's talk. This is the outline of today's talks. I will first talk about how regulatory elements can control gene expression in 3D chromatin structure and contributing to human disease. And in the second part, I will describe in-house developed bioinformatics tools called TENET, tracing enhancer networks using epigenetic traits methods that identifies key transcription factors linked to disease. So we'll start with the first part. As everyone knows here, epigenetics allows cells with the same genome to have different phenotypes. We all from a single human genome, but this single human genome can give rise to many different cell types that have same genetic information, but they have epigenetically different signatures. And those epigenetic differences can lead to phenotypic differences. We study epigenomic features, especially our focus is on regulatory elements, which can lead to gene expression changes and resulting in different phenotypes among many different cell types. Regulatory elements, what are regulatory elements? As you guys know, most common regulatory elements are kind of called promoter here near transcription start sites. So regulatory elements are the DNA regions and that can change the expression of some genes. And promoters are known to be located right next to transcription start sites. However, regulatory elements such as enhancers and insulators are located far from those transcription start sites. Interestingly, those regulatory elements have different activities. Same regulatory elements can be inactive in one cell type, and when they are inactive, there will be no transcript. However, those same regulatory elements can be active, and then they can transcribe the genes. And these chromatin interactions, such as promoters and enhancer interactions, seems to be found, and it's supported by our work and also many other researchers' work that those chromatin interactions can also lead to lots of transcription. We studied those regulatory elements and interactions using many different functional genomic assays, such as histone modification, ChIP-seq, CTCF transcription ChIP-seq, and some open chromatin assays, NOMe-seq, DNA-seq, FAIR-seq, Occupancy. And also we study DNA methylation levels using arrays and the whole genome sequencing. We also study chromatin interactions using chromosome conformation capture, 3C-based assays like GWAS, Capture-C, High-C, and also we use RNA-seq and Eclipse-seq. We, as part of ENCODE and also CycENCODE, we generate lots of data sets working with lots of collaborators, researchers in ENCODE and CycENCODE. And those are the number of data sets we generate as part of ENCODE here. So we generated lots of transcription RNA-seq data in many tissues, cell lines, primary cells, and some in vitro differential cells. We also perform DNA-seq, FAIR-seq, all those toxic data set, chromatin accessibility data sets in those biosamples. We also perform ChIP-seq and DNA methylation, RNA binding, as well as 3D chromatin and replication timing data sets. So if you're interested in using those data sets we generated as part of ENCODE, please go to this website and you'll be able to access all of those data sets. Not only ENCODE, I was able to participate in CycENCODE, which is another consortia. ENCODE is particularly, ENCODE is focused on lots of samples, but CycENCODE is particularly focused on psychiatry disease samples. We also were able to generate ChIP-seq and RNA-seq data as part of CycENCODE. And if you actually go to CycENCODE website, you will be able to find lots of transcriptome, genotype, chromatin, and also some High-C data sets generated in psychiatry disease cells or some neural cells. Let me show some of the example ChIP-seq data set we generate here at USC. This is the ChIP-seq signal for H3K4-ME3. So as you know, A3K4-M3 is known to be enriched in promoter regions, and those are located near those transcription start sites. Unlike A3K4-M3, enhancer signatures, A3K4-ME1 and A3K27 oscillation histone marks are found at enhancer regions. As you can see, A3K4-ME3 signal is different from those enhancer signals. There are lots of signals located in distal regions. We also were able to perform CTCF ChIP-seq, and CTCF is known to be enriched at the chromatin structure loop anchors, and you can see that their signal is quite distinct from even the enhancers and the promoter regions. We also use different histone modification, A3K27 trimethylation for repressed regions, identifying some of the chromatin regions that are repressed. And using A3K9 trimethylation, we're able to find some heterochromatin regions. So we generate those ChIP-seq data sets and also identify some chromatin states for some cell lines and primary cells in here. And we were able to analyze those data sets as part of ENCODE. When we tried to put all those ChIP-seq data and then subset our candidate regulatory regions to promoter-like signature, like promoters, and enhancer-like signature, enhancer, and also CTCF sites, we were able to find that there are some signals like promoter-like signature, which has quite high H3K4-M3, and some of them with little of those H3K27 oscillation, CTCF DNAs, and also enhancer-like signature, which has more H3K27 oscillation, near little proximal, and also there are some enhancer which are very far from those transcription start sites. So we're able to categorize those regions promoter-like signature, enhancer-like signature, and also the region that only have CTCF, and then we checked how many are found in human genome. And interestingly, as you can see here, compared to promoter-like signature and CTCF only and some of like DNAs, only H3K4-M3, there were a lot more distal enhancer-like signatures here. There were over 600,000 of candidate regulatory elements, which are we categorized to enhancer-like signatures. And those enhancers seem to be very distinct across cell types, and there seems to be very cell and tissue type specific. If you see this ENCODE Nature 2020 paper, you will be able to identify some enhancers and also some of signatures which are only found in subset of samples or subset of tissues. Let me just briefly introduce our USC Psych ENCODE project. So Psych ENCODE project, we actually studied schizophrenia. So for schizophrenia patients, schizophrenia patients are known to have some problems with the sense of smells. And we hypothesized that maybe they have different epigenome of olfactory neuroepithelium, and that could lead to those disease or that were affected by some types of cause of those diseases. So we decided to recruit patients with schizophrenia and some of the control groups, and we nasal biopsied them, and then we collected neuronal protein cells and then cultured them and generated C-non cell cultured neuronal cells derived from olfactory neuroepithelium. We were able to collect all of those cells and then chromatin from each individual, and then we performed ChIP-seq as I showed previous slides. This is the result of our ChIP-seq data. We actually performed some of data analysis using subset of individual after quality check. So A3K4-M3, we were able to develop this ChIP-seq data from 56 individuals, and we checked the ChIP-seq signals at the peaks which are found in all individuals, 100%, and peaks which are found more than 20% of individuals and less than 20% individuals. And as you can see, A3K4-M3 mark seems to have a lot more strong signal for the peaks that are found in all individuals. And if you see 20% and less than 20%, the signal was actually very small. When we look at the CTCF, we also found that the CTCF ChIP-seq peaks, which are found in all individuals, had a high signals, but there were some signals which seems to have a lot more compared to this H3K4-ME3 pattern. We see some signals which are still stronger that are found in subset of individuals. We also did H3K27 oscillation ChIP-seq, and as you can see, H3K27 oscillation ChIP-seq signal was very high for the peaks that are found in all individuals, but there were still some peaks which are found in more than 20%, and their signal level was not was quite strong, unlike H3K4-ME3. Next, we checked how many H3K4-ME3, CTCF, and H3K27 oscillation ChIP-seq peaks are found in common or unique for each individual. So we generate these bar plots. So the x-axis shows that the peaks, if peaks are found in only one individual or 20 or more than 50 individual here, and the y-axis shows the number of peaks here. And as you can see, when we did analysis using H3K4-ME3 ChIP-seq peaks near transcription start sites, we found that a lot of H3K4-ME3 peaks near those TSS sites promoters are found in all individuals. However, insulators and enhancers, which are located distal sites from promoter regions, seems to have some peaks which are not found in all, but in a subset of individuals. And the numbers are like 29,000, and in enhancer cases, we found a lot more, 56,000. And actually, you can see here, compared to insulator and enhancer signal, there are a lot more stronger and more enhancers which are found only in a subset of individuals. So we can see that enhancer activity is most variable across individuals for a given cell type. We did this assay in one cell type, xenon. Not only enhancer activity is individual specific, we were able to find some disease specific enhancers. As I mentioned, for Psych-encode project, we were able to recruit people who are diagnosed with schizophrenia and also control groups. And we performed analysis between those groups, and we identified some of ChIP-seq signals found only in disease schizophrenia groups. For example, near NCF4. We did not only study for schizophrenia, but we also study other cancer like prostate cancer, and using some cell lines, we were able to find prostate cancer specific enhancers, and also we study some breast cancer enhancers using MDM231 basal breast cancer cell lines and human memory epithelial cell normal breast cell lines, and we were able to find some normal breast specific enhancers as well as breast cancer specific enhancers. Okay, so we now know that among regulatory elements, enhancers are very cell type, individual specific, and also disease specific. Then why they are very specific in disease or cell types, right? And we want to study what causes those and realize that those are where transcription factors bind. However, interestingly, H3K27 oscillation signals seem to have quite broad here and sometimes up and down, and transcription factors are not necessarily bind to those middle of H3K27 oscillation peak here, but transcription factors actually bind to where this dip is, where those signals goes down here. And we realize that those regions are nucleosome depleted regions. And those nucleosome depleted regions we call NDR can be identified by using chromatin accessibility assays such as DNase, FAIR, and ATAC and NOMe-seq. So we wanted to study what those transcription factors bind to these each cell type disease specific enhancers. To do that, we performed some chromatin accessibility assays, and such as DNase, which is DNase digestion, and then find some of the regions which are open. We also use mRNAs, which can digest more, but unlike DNase, we can see the signals inversely. And FAIR-seq can be also performed by using sonication, and then we are able to find open chromatin regions. And relatively more recent assay, ATAC-seq can be performed to identify some open chromatin regions here. So we performed those assays as well as another assay called NOMe-seq, Nucleosome Occupancy and Methylome-seq, which is actually developed here at USC by Peter Jones' group. So what is NOMe-seq? So NOMe-seq is another way to identify some nucleosome depleted regions, but we are not using some of fragmentation or toxic transposon, but we use the DNA methylation methyltransferase GPC. So basically, NOMe-seq actually can identify endogenous DNA methylation, also GPC sites. So how NOMe-seq works is that we isolate nuclei and then we treat with MCVI-PI methyltransferase that can put methyl group site, and those regions are open, GPC sites can be methylated. And actually this MCVI is isolated from cholera virus, and it's not GPC sites are not methylated in human mostly. Therefore, we're able to use those enzymes to find nucleosome occupancy profiles and some nucleosome depleted region. So we isolate nuclei and then we treat with this MCVI. The regions that are open will be methylated, GPC methylated. After we get those, we can perform bisulfite conversion and then sequencing to identify some nucleosome and also endogenous DNA methylation. So as I mentioned, NOMe-seq has lots of advantages and pros. It's not reliant on some histone modification, or it's not using the DNA practice sonication or DNase or transposon integration. So you'll be able to map the nucleosome more naive way, and you're able to find some position information using this NOMe-seq because it will tell us several nucleosome occupancy regions near open chromatin region, like such as enhancers. Also, NOMe-seq has advantage because we use endogenous DNA methylation, and using this CpG GPC DNA methylation, we'll be able to find precise percentage of those openness, and also we can map single molecule resolution. You'll be able to see more of NOMe-seq advantage from these papers. So we performed this NOMe-seq in some of cells like xenon and also some process cells. And this is the example of our NOMe-seq data. We did the NOMe-seq and then we found some nucleosome depleted regions which have high GPC because they are open. GPC sites are methylated. So this shows that DNA methylation and accessibility percentage. So you can see that at NDR, the GPC methylation level is high here. And the endogenous DNA methylation, CpG methylation level, is relatively low here. And you can see that when you look at here on the right side, chromatin accessibility and methylations are shown, and it's very single nucleosome molecule resolution. And we found that after generating these heat maps at all those NDRs, there were some NDRs which tend to have like quite wider open regions and the smaller region. And we realized that enhancer seems to have smaller NDR sites with accessibility and a little less CpG methylation here. So we identified those NOMe-seq, and then we went back to our H3K27 oscillation ChIP-seq data, and we decided to find some nucleosome depleted regions, and then perform some motif search, and found that for xenon cases, olfactory neurons, we were able to find the transcription factor motif of NFE2 motif is enriched in these olfactory neuron specific enhancers. However, when we perform the same assays in other cell lines, for example, process cell, cancer cells, we found that different transcription factor motif is enriched. For example, here, FOXA1. And we were able to actually perform FOXA1 ChIP-seq to confirm that FOXA1 binds to these nucleosome depleted regions in these enhancers. So using all of those ChIP-seq and MDR from NOMe-seq and motif search, we'll be able to find transcription factors that specifically bind to those enhancers. Okay, now we are able to identify cell type specific enhancers and key transcription factors that bind to those regions using this approach. However, there is another question, because enhancer is not necessarily regulate the nearby gene, but it can regulate genes that are far from those enhancers. So to answer those, we perform some chromatin conformation capture assays, genome-wide chromosome conformation capture in situ. So in situ Hi-C is a method that's developed by other groups that can check the chromatin interactions, and you can identify some of the promoters and enhancer interactions. And we perform those in situ Hi-C in process cells, for example, RWP1 normal process cells, and then 212 prostate cancer cells. I will tell you how to read this heat map. So this is the Hi-C heat map. So if one region interacts with another region, and if it interacts a lot, the color of this here heat map is going to be red. But if one region does not interact with another region, then the color is going to be whitish color here. So we developed this in situ Hi-C high resolution maps in normal prostate and then 212 prostate cancer cells, and then we visualize those chromatin maps near androgen receptor, which is known to be involved in prostate carcinogenesis. And we found that chromatin structure, chromatin interaction maps are very different between normal and prostate cancer cells. As you can see, near androgen receptor for prostate cancer cases, there are like smaller TADs, which is actually topologically associating domain. So you can see that chromatin interactions are found more frequently near those androgen receptor for prostate cancer cells, but not that much for RWP1. So we wonder what's going on here, and then we overlaid with H3 RNA-seq and A3K transcription ChIP-seq data, and we found that there are some androgen receptor AR specific enhancers that are only found in cancer cells here, and they tend to be associated with the RNA-seq expression changes. So using Hi-C, we're able to find some enhancer promoter interactions, and we were able to find those chromatin interactions and maps. So many cancer-specific enhancer promoter loops near the androgen receptors are found, and we think that this enhancer promoter loops can result in overexpression of androgen receptor for these cells. Okay. There are many ways to study a disease, but not only just using ChIP-seq, TAC-seq, and NOMe-seq or RNA-seq, but some people are studying disease using genetic information. So genome-wide association studies are another studies that they study genetic factors that lead to disease. So how GWAS works, genome-wide association study works, is that they recruit many individuals who are diagnosed with disease, like for example, prostate cancer, and they also recruit control groups, and then we sequence their genome, okay, using arrays or sequencing, whole genome sequencing, and then they compare the SNPs, single nucleotide polymorphism, and then they find some of the alleles that have different frequency. For example, here, SNP1, same SNP, but people with these cases, like for example, prostate cancer, they tend to have more A allele compared to this control group, which have more T. And by performing some analysis, we're able to find some SNPs that are associated with risk. So we are able to work with epidemiologists here at USC, and we found some of the risk SNPs that are associated with prostate cancer and breast cancer. However, there's a big dilemma for GWAS, because when we found those SNPs, their location is not in coding region, it's not in gene, but it's located in non-coding regions. When we performed analysis using the gene information, you can see that a lot of them, not only for prostate, but also many breast, lung, and other cancer types, many GWAS-identified SNPs are located in intergenic or internal region. But actually that's true, that makes sense, because as you guys all know, our human genome has a lot of part, more than 98% of them are non-coding regions. Only 2% of them are protein coding genes. So definitely GWAS people need our help to understand the cause or the function, how mechanism of how these risk SNPs lead to disease. And we actually participated this study working with epidemiologists, and we studied on prostate cancer. This is how we study prostate cancer risk SNPs. we are able to find prostate cancer risk SNPs at distal regulatory elements first using 2000 fine mapped prostate cancer GWAS SNPs. After performing GWAS, we fine map to narrow down some potential SNPs related to the risk, prostate cancer risk. And then we overlap with the DNA-seq signals, and then we are able to find some SNPs in open chromatin regions like 443 SNPs here. Then we overlap with some enhancer signatures, H3K27 acetylation, and then CTCF signals here. And then we were able to narrow down from 443 to 222 SNPs, and then 93 SNPs located at CTCF sites. And then we brought our high-C looping data and narrowed down to the 201 and 85 SNPs. And as I talked about H3K27 acetylation enhancer history on the previous slides, I will talk about some of the SNPs we studied that they are located at CTCF sites. So among those 85, we studied a little further, and one of SNP is called RS1214497A. This SNP is located in this site 1 we named, which is CTCF site here. And when we look at the chromatin interactions map, we found that this site 1 interacts with another CTCF, site 2 and site 3 here. Okay. And this site 1, this risk SNP is located in non-coding regions and there are no genes. So we wonder what gene is regulated by those CTCF sites, this SNP. To check that, we decided to perform CRISPR-Cas9 experiments, and we deleted the CTCF sites here, whole chunk, and then performed RNA-seq. And interestingly, as you can see, the KCNN3 gene expression level actually went up more than 100-fold when we deleted site 1, which was very striking. And we asked, how about if we delete another site 2 and site 3? So we deleted site 2 and site 3. However, we didn't see the expression change for KCNN3. However, when we deleted together with site 1, we were able to see the very strong signal changes. We wanted to know what's the molecular mechanism of this process, and then we overlaid with lots of other ChIP-seq data, and we realized that actually this chromatin loop is heavily enriched with repressive mark, H3K27 trimethylation mark. You can see here, this is the location of our SNP, and CTCF site 1 and site 2, site 3, it makes loop, and then it has very high enriched H3K27 trimethylation. We wonder whether there are some enhancer signatures, and then we found that there's one big enhancer on the right side here. And we wonder how deletion of this site 1 only can lead to this gene expression change up to increase the expression up to 100-fold. So we decided to perform ChIP-seq after deletion of these sites, and then we realized that there are not new enhancers heavily found, heavily enriched. There were signals for the promoters, but not for the enhancers. And we realized that this enhancer still has a very high signals here. So using those data set, we came up with this model. So when this CTCF is there, when there's no deletion, CTCF loops, repressive loops are very strong. So this enhancer cannot access and cannot increase the expression of KCNN3 here. However, when we delete this site 1 here, this enhancer can go in and access to the promoter, and then it increase the KCNN3. So that's how we came up with this model, and then we actually performed some of the studies in other regions, and then we showed those that repressive loops and enhancer activity are changing those gene expression levels here. So in summary of this part 1, we built as part of ENCODE and PsycENCODE over 1 million functional regulatory elements in non-coding regions. And we are able to identify individual and cell type specific enhancers in various tissues and cell types. And we were able to identify enhancer NDRs using NOMe-seq And we were able to find some cell type specific transcription factor bind and then work on those enhancers. And using Hi-C data, we were able to find the target genes of those enhancers and integrating GWAS SNPs and integrating with ChIP-seq, NOMe-seq, and Hi-C those data set, we were able to find some of the target genes of those SNPs. And by integrating data, we were also able to find some molecular mechanism behind those SNPs. Okay, so let's go to the next part. So first part is done, and I will briefly talk about our bioinformatic method, TENET, tracing enhancer networks using Epigenetic Traits method, which can identify key transcription factors linked to disease. I participate TCGA, as Jerry introduced me. So what is TCGA? So TCGA is one of the largest genomic consortia that we profiled epigenome and genome and transcriptome of lots of tumor tissues. And I was here at USC, I'm here at USC, and I worked with many people here, Johns Hopkins and other here, by collecting many tissues. And I was one of the leaders who studied DNA methylation. So TCGA data actually are generated from tissues, and actually performing ChIP-seq and TEC-seq, all those NOMe-seq epigenomic data is very challenging. However, profiling DNA methylation is relatively easy because DNA methylation can be done using just the DNA, even from FFP samples. What is DNA methylation? So DNA methylation is usually endogenous, we refer to as endogenous CpG, and cytosine can be methylated when it becomes methylated here. And this CpG DNA methylation levels can be inferred by using bisulfite sequencing, which can convert just a C to U, but if it's methylated, it's not going to be converted, so we're able to distinguish the DNA methylation levels. We performed bisulfite sequencing as well as array as part of TCGA, and we generated many DNA methylation data sets in hundreds of tumor tissues. And we found that DNA methylation levels can be used to infer open and closed chromatin states. So for example, one gene here, this is the promoter regions, and enhancer regions, we realize that when promoter regions and enhancer regions are methylated, they tend to have no gene expression. However, when they are unmethylated, open circle, we were able to see that the gene expression levels are high. We were able to confirm these changes between normal and tumor using TCGA data here. So this scatter plot, each dot indicates each sample. The normal samples are colored with this blue here, and tumor samples are colored with reddish color here. And as you can see here, this is the near GSTP1 promoter regions. And for tumor cases, they have high DNA methylation level, and they have relatively lower gene expression for GSTP1 compared to normal. Not only for GSTP1, we can see that KLF8, the hexa and STAT, and also over 100 genes, we found that hypermethylation at those promoter regions lead to the gene expression silencing. We confirmed that those expression changes are not related to copy number variation, somatic mutation, or even tumor priority by visualizing with different shape. And then we confirmed that this epigenetically changing DNA methylation level lead to expression changes. Not only promoter region, but also enhancer regions can change the gene expression, and DNA methylation levels at enhancers can be used to infer enhancer activity. As you heard from my part 1 study, there are some cell type specific enhancers, and we know that key transcription factor like cell type, distinct sets of transcription factors bind to those enhancers. And we had one big scientific question: is it possible to find some key transcription factors that are linked to cancer type specific enhancers using TCGA data sets? And TCGA data set, as I mentioned, are mostly done from tissues, and we couldn't perform ChIP-seq or TEC-seq or NOMe-seq those assays, just DNA methylation. And can we identify key TFs using those TCGA data? And we try to answer those questions and by developing our method, TENET, tracing enhancer networks using Epigenetic Traits. So this is the hypothesis behind, the logic behind TENET. So there's one transcription factor, and this transcription factor expression level can be low, and the protein level can be low. And since it's low in here, for example, in normal, it binds to only a small number of enhancers and regulate the gene, only small gene. However, same transcription factor in tumor can be activated, that can be oncogenic, and then it expresses a lot more protein, and then they can bind to a lot more enhancers and then lead to overexpression of many oncogenes. And we want to find those sets of transcription factors that are activated and then bind to cancer-specific enhancers and regulate cancer-specific genes. And that's how the logic, and then we were able to develop our tool TENET. So how TENET works is that instead of using enhancer open chromatin data from tissues, you can use the TENET database, or if you have some ChIP-seq data from related cells, you can include those and then identify DNA methylation CPG sites probes located in enhancer regions. And TENET, next step, compares DNA methylation levels between normal and tumor and identifies some differential DNA methylation sites that includes the hypomethylation sites, which we mean the tumor-specific enhancers. And then we use the RNA-seq data from matched samples. So most TCGA data have matched RNA-seq data. And using those, we are able to identify some transcription factor whose expression levels are associated with DNA methylation changes of those enhancers. And TENET perform those assays, whole all genes and all enhancers, and identify key transcription factors that are linked to many cell type specific enhancers. And then it also uses high-C and genomic and clinical data sets to perform downstream analysis to help understand the transcription factor enhancer gene networks. So using those input data set, we'll be able to identify differentially activate enhancers, key transcription factors, and also some tumor subgroup enhancer expression analysis activities, and some key TFs and patient survival, and also all network. I will describe how we perform these assays for one by one here. So we use the ChIP-seq and NDR data from some cell lines and some of samples, and then we overlap with the CPG sites that are located far from the transcriptor site enhancer probes. And then using DNA methylation data set, for example, breast cancer data for TCGA, we were able to compare the DNA methylation between normal and tumor and identify like over 19,000 of enhancer probes that have hypomethylation. So that means that in tumor, it's unmethylated, but in normal, it's methylated. And we confirmed this enhancer signature with some whole genome biosuppress sequencing data. So for example, this enhancer here, CG04747693, activity is open only in breast cancer. You can see that unmethylation is found in breast cancer and actually in luminal A and HER2, but not in basal, not in normal, and also not in other cancer types. So we were able to find those cell type specific enhancers comparing DNA methylation data from tissues using TENET. And then TENET identifies key transcription factors linked to those enhancer activity. As other and also previous studies show that DNA methylation level for the enhancer regions are related to the transcription factor gene expression for the regions that are bound by this transcription factor. So we compare the DNA methylation levels and the transcription factor gene expression levels, like making this scatter plot, and found that some of enhancers and transcription factor gene expression is linked like that. So for example, tumors have low DNA methylation, but have higher gene expression, for example, HOXA6. And we perform this analysis to all enhancer and all TF, and then we generate thousands of those scatter plots and analyze it. And then we combine the analysis, and we found that a subset of transcription factors, very small set of transcription factors, are actually linked to those cancer-specific enhancers. And when we visualized, we generate circus plot here. And for example, GATA3 for breast cancer is known to be looped to many cancer-specific enhancers here. And we found that not only GATA3, SPDF, FOXA1, ESR1, estrogen receptor, which are known to be very important for prostate breast carcinogenesis, are identified. And we also performed this analysis in another cancer type like prostate adenocarcinoma, and we found that different sets of transcription factors are found, like HOXA6, HOXA4, and DLX1. We realized that those transcription factors are known biomarkers for prostate adenocarcinoma. We did develop TENET 2.0 using more advanced algorithm integrating survivor. And we found that some key transcription factor for lung adenocarcinoma, for example, MIBL2, and MIBL2 expression level was much higher. And then for the tumor samples which have higher expression of MIBL2 seems to be associated with a poor survivor. And as I showed in the previous slide, MIBL2 is actually linked to many of lung cancer specific enhancers. We're able to integrate our TENET 2.0 data using Hi-C data and identify some target gene of those enhancers. For example, MIBL2 linked enhancer CG09580922 is linked to those TK1 gene, and this TK1 gene expression level is also associated with poor survivor high, and also enhanced activity is also associated with poor survivor. So using those TENET 2.0, not only the transcription factor important for disease, but also associated with survivor can be driven, can be identified, and target genes of those enhancers can be identified. So TENET 2.0 is available in GitHub page here. And if you go to this GitHub here, you will be able to find how key functions work. So to summarize it, we identified thousands of activated enhancers in tumor tissue samples, and using just DNA methylation levels and some RNA-seq data from tissues. And we developed TENET and identified some key transcription factors that are uniquely found in each cancer types. And TENET 2.0 can use patient survivor data and also some target gene data, so you'll be able to find some key enhancer signatures. I'd like to thank people, especially Dr. Farnham and some of her previous former postdoc and also PhD students and some technicians who generate lots of ENCODE and psych-ENCODE data. We worked with the Peter Layer, Peter Jones, and Dr. James Knowles for the psych-ENCODE. And also we have other collaborators, Dr. Fringa and Daniel Miller, who developed the TENET 2.0 with me, and also Shannon, who developed lots of data. And we have some fundings here from NIH and DoD and others. And lastly, I'd like to highlight that I am an assistant professor. I opened my lab recently, and we are looking for some talented students or postdocs. So if you're interested, please contact me through email or Twitter. And lastly, but not leastly, USC Keck School of Medicine here in our department, Biochemistry and Molecular Medicine, has very well organized graduate programs. If you're interested in it, please apply using this website. And if you want to just talk about your career, feel free to reach me. Thank you so much for your attention.